Hello, everyone. So my name is Eric Sid. I'm a program officer within the Division of Rare Diseases Research Innovation at the National Center for Advancing Translational Sciences, or NCATS, at the NIH. And I just wanted to welcome you all for joining us for a session today on the Oligonucleotide Toxicity Open Data Challenge, what we like to call too as the OligoTox Challenge. So before we get started, I'm just going to go ahead and give a quick introduction to the agenda today, as well as some of our different speakers. So hopefully this is starting to go. There we go. We're going to be going through and talking a little bit about the challenge, its goals, its background, as well as some information that's going to be detailed both in brief here, but a lot of this will be referring what's actually posted already in challenge.gov, as well as on NCATS's page on the challenge. We're going to talk a little bit about the schedule, the prizes, the submitting, as well as evaluation and judging. And we really wanted to spend the majority of today focusing on any questions that you might have. The three of us that will be speaking today include, again, myself. I'm Eric Sid. I'm a program director. PJ Brooks, who's our acting division director, again, for the Division of Rare Diseases Research Innovation. And then one of my colleagues, Element, she's also on the line. She's our acting extramural research operations officer, assisting us with this challenge. So I would just ask, as we're going through, if you can just hold your questions until the end when we have a dedicated Q&A. And as we're going through, if there's anything that you'd like to bring up, you can go ahead and put that into the Q&A within the Teams app itself. So, and again, I apologize, I can talk a little fast, so I'm going to try to slow myself down. The OligoTox Challenge, just to give you some background behind it, oligonucleotides, or oligos, as we like to refer to them, are a class of therapeutics that have been an increasing number of drugs in development, as well as several have been approved for use over the last several years recently, in particular for the treatment of various rare genetic diseases. Now, over the last year in particular, the FDA, the Food and Drug Administration, as well as the NIH, National Institutes of Health, are both interested in what's referred to as new approach methodologies that can be used to reduce the use of animal testing in preclinical safety studies, such as through technologies like organoids, which are small lab-grown tissue models that can replicate the structure and function of human organs. As well as in recent years, many of us have obviously witnessed the dramatic increase in the use of advanced computational methods like generational AI to address complex biological problems as well. In that setting, we proposed this challenge. NCATS is seeking to incentivize the generation of high quality and public accessible data sets that can make use of in vitro human systems and can be used to advance predictive models for oligotoxicity. In particular, we're interested in toxicities such as hepatotoxicity of the liver, nephrotoxicity of kidney, thrombocytopenia, complement activation, coagulopathy, immunotoxicity, chronic neurotoxicity, and hydrocephalus. But again, not limited to these toxicities only. This challenge as a whole has two phases to it. The first phase, which is what we're going to be primarily focusing on with this webinar, is the ideation phase. Now, it already started on December 19th and is going to be going until the end of February for submissions. And the aim of this phase is for proposing how you would collect, build, or contribute a data set from in vitro human-based systems that can be used to support the development and training of in silico models that can help accurately predict toxicity for a candidate oligotherapeutic. Again, the intent of this one is really to help us spark ideas for what types of data sets would be vital in, again, crafting these types of in silico models that are necessary for, hopefully, being able to advance the field of oligotherapeutics. The prizes that we're going to be offering for this first phase are going to be limited to a maximum of 10 winners with a prize value of a purse of up to $10,000 each. The second phase, which again we'll have a separate webinar for in May when this is officially starting, will be held starting in May and last the rest of the year. So this is going to be a lot more time for phase 2. Phase 2 is meant to basically take off after its ideation phase. This would be to actually make these data sets themselves publicly available, high quality open data sets, and have demonstrable use in developing and training predictive models for oligonucleotide toxicity. The prizes for this are, again, a higher level. There are going to be a maximum of 2 winners with a prize of up to 100,000 each, and up to 4 runners-up with a maximum prize of up to $50,000 each. There are some caveats. If we do not receive enough applications for phase 1, we may reallocate some of that the funding to phase 2 winners. And again, this is all detailed out more in detail in the posting and the challenge itself. And we have a link to that in the last slide in case you haven't seen it yet. Now, again, this is a very detailed slide with a lot of notes. I'm just going to cover a selection of this. Please refer to the listing for the submission requirements in detail. For phase 1, we're really looking for a description of how the proposed data set would be collected, generated, or provided, where you're basically intending on pulling the source of data from, as well as the suitability of the data for this challenge. We want to understand that you have a good understanding of the indicators and predictor variables that would be used for evaluating the toxicity of oligonucleotide, as well as the evidence and references that you're using to supporting that these indicators have reliable and valid use in predicting toxicity. And some of the descriptors, as an example, that we're looking for are model systems that are being used, toxicity readouts that are assessed, estimated number of oligos that have been evaluated, as well as any positive or negative controls, methods that were used for purifying oligos or oligo concentrations and doses, as well as the number of replicants. The evaluation and judging itself will go through a first review with assessing for if the submissions comply with eligibility and submission requirements as a whole. Those that do are going to be scored by a technical panel for scientific value, experimental approach and design, data translatability, as well as proposed data set management. Finally, a judging panel will take these scores as well as be reviewing these specifically for their confidence rating on how well submitters address these factors. The ability to solve the challenge itself, whether or not this is appropriate in advancing the issue of predictive modeling and oligotoxicity. The potential impact this might have. Is this data set addressing important information gaps that otherwise aren't being addressed at this current point? The feasibility and rigor of the data set, whether or not the viability and quality of the data set suggests researchers would be interested in making use of it, as well as the transparency and reproducibility of this data. And we've pointed to NIH scientific data sharing policies and best practices as an example. And again, I just wanted to note some of the important dates here, but these are all listed in the challenge posting. Phase 1 submission has already started and will go until the end of February. A winner will be announced at the end of April. Phase 2, which we will have a separate webinar itself to go more into detail on, will be in tentatively mid-May, and will start in May and last through the end of the year. The winners for that second phase will be announced by March of next year. So with, again, that very, very quick overview of a much more extensive, detailed posting on challenge.gov as well as on the NCATS website, I now wanted to really open up this to questions and answers. And again, I wanted to look to my colleagues, PJ as well as Elmid, to join me if there's any questions that folks in the audience want to bring up. William, go ahead. I see that you have your hand raised.
So I just have a question. So in the application, when you say certain toxicities will be given a lower priority, how does that work in the overall scoring? Does it mean that they would only be looked at if there were no submissions of the other toxicities, or would it be a reduction, like some type of weighted reduction in the actual scoring of the application?
We would anticipate that would be sort of a weighted reduction.
David?
Thanks, Eric. I would like some information about how you plan to administer these oligos. Are these going to be administered directly to the brain? Or are you going to put them and inject them into blood vessels with some kind of delivery vehicle? Because that will maybe be important information to figure out what the toxicities are that we expect.
So, David, the focus of this is really be able to ultimately generate data from in vitro systems that would be relevant to toxicities. And so thinking about the ultimate, like we're not supporting any actual treatments here or actually administering oligos. It's really just to generate toxicity data, as Eric said. But obviously one would want to consider the ultimate goal at administration in trying to design the toxicity studies.
Yeah, so I was part of a very large project in large pharma where we were looking at systemic delivery of siRNA molecules and delivery specifically to liver hepatocytes. And so under that kind of goal in administration, the toxicities, the dose-limiting toxicities were all about the delivery vehicles. And so I think in order to address the question here, we kind of need some information about how you would like to use this to develop therapeutics. In other words, are these going to be administered to the brain, as Don Lowe wanted to do, directly, or are they going to be administered, systemically and to target different organs?
Well, David, I just want to mention there was a recent paper that came out that showed a siRNA targeting the some type of, I forget the exact kidney disease, but actually they found neurotox with that because it surprisingly crosses the blood-brain barrier. So I just want to say a lot of these oligonucleotide toxicities, they can be, like you're saying, they can be widespread depending on how you're administrating them. And, so I do think that is a important point to kind of take into kind of a general consideration.
Yep, I would agree.
Okay.
And one thing I'll just note too is that I think part of what we're trying to do with this effort is really focus on the preclinical space of this as well, because part of the question we have here is, are there basically gaps in terms of the availability of some data in the public sphere that could help with improving the processing of, like, for example, oligonucleotide toxicity at preclinical in the preclinical space more quickly with either models that could be shared more to the field as a whole. The problem being that a lot of the times the data that they're using to train their models isn't necessarily available to everyone else. So part of this is seeing if we can both, in addition to getting some more exposure to those models in the future, see if we can start to build more of a publicly available data set that is used to develop and train these types of models for preclinical toxicity prediction.
Let's see, I think a couple other people have joined since we started. I do apologize to some of you that because of the challenges around the government shutdown, et cetera, there was some delay in getting to the mailbox. So I sent out some of the links quite late today. And so if anybody has any questions here, even about some of the stuff that you may have missed, please go ahead and raise your hand. But I guess also, I mean, if not, and there are no questions, Zach?
Hi, yes, thank you for having this webinar. And I just had a general question, kind of, to the point about, I mean, is there any preference for the type of oligo, siRNA, like data sets generated from siRNAs or ASOs? And to that end, like let's say, for instance, ASOs. We recently published a paper in Cell where we looked at postmortem and biofluids from an ASO therapy that failed in clinical trial against C9orf72 and found a number of human-specific changes. But that was just one ASO. If we were to study ASOs, would you want multiple different ASOs tested? Because we're seeing even now that multiple different ASOs cause very different immunogenic responses, even if the modifications are the same. And so I guess, would it, maybe we can't test, 1,000 different sequences, but we could take, a number of FDA approved or preclinical ASO sequences and test those. Just kind of get an idea of, what you might be looking for.
I guess in the long term, I'll just take this and then you can hop in. What we'd like to do, we really would be able to ultimately predict toxicities without using animals. So right now, as you mentioned, we can't. There's a lot of data that we don't have. But what we're trying to think about is what would be the most valuable data that could be generated that we would use to feed large language models, other types of models to be able to predict those toxicities. Does that help? Yeah,
no, that's very helpful. And what if we already have these data sets? Can we enter that into the, we have large data sets already collected from biofluids of about 300 CSF of ASO treated patients for neurological diseases.
Ultimately, you want them to be able to put into the public domain, but
yes,
you certainly could.
Yeah.
Okay.
Yeah, this is why we emphasize it's not just the generation of new data by collecting it. It could be the contribution of existing data, contribution of data that may come from previous trials or even failed trials. What we're really hoping for is to just get a better understanding of what data could be made available to really help the development of these types of predictive modeling approaches.
Okay. And one thing on the prizes, I was just interested, is this something that it's not a grant, right? Who does the prize go to? The university? I mean, if you're at a university.
Element, do we have that information? I've done one of these before. I believe that it actually goes to the individual who submits, but I will look into that and we will post.
Yeah, that's a good question. We will double check with grants management and provide you the information.
Okay.
I don't want to mislead anyone. Yeah.
Yeah, because you're right.
Thank
it is not a grant. It's literally a prize.
It is a prize, yeah.
Yeah, and so I saw where there was a comment that's like, oh, you'd have to pay. There's, you got to pay taxes on it, right? So that's why I was confused if it was going to the individual or going to the university, and so I didn't know if we needed to submit this to our university as like we're submitting something, right?
I think that you'd want to check with a university, but typically not. That, like I said, this doesn't go through the typical NIH grant submission process.
Okay. Yeah, the one thing I was thinking about also is that what would be a little different for phase 1 versus phase 2. Phase 1 is really an ideation phase. So phase 2, if you're going to be submitting the data itself, again, that would go back to then who owns the data.
Okay. Yes. And yeah, I mean, that always gets complicated, right, with universities. But
yes.
The next question is for the ideation phase. Can we submit 2 different proposals or ideas?
Yeah, I don't think we put a limit on how many submissions we could come from individual people.
Okay.
As far as I'm aware.
Thank you.
Yeah. It's like Jyoti.
Yes, I have a question. So I'm not sure like who, if you're applying like individually, who can apply, because I'm a postdoc. So am I eligible to apply? That part was not clear.
Yeah. So if you looked at the posting, it will tell you you can apply as an individual. You can also apply as a team. If you apply as a team, you usually I think you have to have a spokesperson. There are specific eligibility requirements in terms of, I believe, like you have to have at least somebody that's, I believe, a U.S. resident. But it's all detailed out more in the posting itself on Challenge.
Okay, thanks.
Element, did you want to add to that? Oh, you're on mute.
Yeah, I just want to mention that the eligibility criteria for who can apply and with the submission are indicated specifically and very detailed in the on challenge.gov. If you're not sure and you have particular questions, you're welcome to contact us and we will guide you accordingly.
And Wednesday just posted the link in the chat text today.
Yeah, thank you.
I'm not seeing any more raised hands or any more questions. So if we don't have any, we can certainly end early. We will be posting this recording as indicated there on our website, so people can refer to it. And we also have the box for questions. And again, I apologize for missing some of these for sort of technical reasons, but we will certainly be doing a better job of monitoring these box for questions, and happy to follow up with anybody who has them.
There was a question whether the slides will be shared in the Q&A.
We can share that with our communication staff, and we'll see if we can get that up.
Yep.
Any remaining questions from anyone else? Thank you for your interest. Thank you for joining us, and we look forward to your submissions.
Thank you.
Thank you.